Hello, everyone, and welcome to today's webinar, Gene Inactivation by CRISPRi Modulate Gene Expression with PAM-anchored Targeting. I'm Alexis Cross of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Horizon Discovery. To learn more about our sponsor, please visit horizondiscovery.com. We encourage you to participate today by submitting as many questions as you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Steve Smith, a product manager for gene modulation at Horizon Discovery. For a full biography on our speaker, please click on the Biography tab at the top right of your screen. Steve, welcome, sir. We're happy to have you. You may now begin your presentation. Thank you, Steve, for that wonderful and informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So, Steve, let's get started. We've already got some amazing questions coming in from the audience. So we'll start with this one. Now, this first one is going to be a two-part question. Do you see any increase in off-target repression with the more potent repressor? And the second part is, would CRISPRi with pooled synthetic sgRNA be less specific than using an individual guide RNA?
Oh, right. Yeah, Alexis, thanks. And thank you for that question. This is something that as we've been sharing this with folks a little bit, through the development, this has come up a number of times. So thanks for asking that in the forum here. In order for a off-target effect, in order for a guide to have an off-target effect, it would have to be an off-target binding to another regulatory region. So that really shrinks the space for off-targeting. We have definitely performed RNA-seq on our data sets to compare the specificity of our repressor to the crab repressor. We found that there's just really very, very minimal bystander effects resulting from using these guys. We did look at the pooling of guide RNAs versus individuals as well, and really didn't see any increase in differentially expressed transcripts. Yes, I mean, we definitely offer the pools and individual reagents so you can follow up on correlation and address any off-target concerns. The other thing that's come up a bit is whether there's any, truly non-specific actions as far as, acting on next door neighbor genes or across the street neighbor genes. And we just really, because of that limited window for efficacy where these guides need to be placed in relation to the transcription start site, we really don't see any evidence of these effects, though it's a reasonable concern and something we're certainly keeping our eye on. So I think the beauty of the system is the specificity in large part, and you have to have kind of prolonged contact for that CRISPRi mediated repression. So it's cool. Thanks for the question.
Of course. So moving on to our next question, you mentioned the time course for stably transfected cells. How long do you expect the repression mediated by dCas9 Sal1 SDS3 mRNA to persist for?
Yeah, right. Yeah, thanks. I might not have mentioned that in passing. So we see really robust transcriptional repression that comes on very, very quickly as you're giving it kind of a large bolus of reagents. And then this, we recommend this approach for those short endpoint assays in the 2 to 4 day range. I'd say is the safe window to be looking at when using the mRNA along with the synthetic single guides.
And continuing on with repression, what level of repression should I expect to see with CRISPRi, and how does the level compare to siRNA effects?
Yeah, sure. So, this is the level of transcriptional repression with CRISPRi is going to, like we're dealing with these pesky biological systems, right? So you're going to have some variance depending on your gene target, cell line, time point of analysis, et cetera. We definitely see, comes online quickly. The target gene repression is maximal in the 48 to 96 hour sort of window, and we've seen that 50 to 80%. I called it out, 60 to 80% knockdown during that time. So this is definitely somewhat less than we observe with our siRNA reagents. We expect those usually in the 80 to sort of 95% knockdown area.
Great. Now, our next question. Is it possible to use CRISPR and CRISPRi together for activating a gene and inactivating another one at the same time?
Yeah, wow, cool. Yeah, so kind of looking at simultaneous activation and interference. There's some technical hurdles there as far as the guide design and controlling which genes to turn on and which genes to turn off when mixing them all. So at this point, no. And at this point, we're not able to do simultaneous switch on, switch off across various gene targets. But it's a super cool idea and something certainly I'm sure us and others are keen on and seeing if there's a way to accomplish that, because that would be super cool.
And so our next question, Steve, do you have the CRISPR mod CRISPRi as an inducible system?
Right, we don't have an inducible flavor of this at this time.
Great, thank you. So we do want to just remind our audience, we've received so many great questions in today. Any questions that we don't have time for or unable to get to today, they'll be answered by Steve or a member of Horizon Discovery via the email address that you registered for this webinar with. So Steve, let's move on to our next question. How are the synthetic guides or synthetic guides plus dCas9 Sal1 SDS3 introduced into the cells? Is it, for example, nucleofection?
Yeah, so thanks. I might not have mentioned that well in passing, but we've done both lipid transfections and nucleofections with the mRNA plus the single guides. So whatever your cells would tolerate the best will work.
Wonderful, thank you. Now, so for the multiplexing of many sgRNA against different targets, was that done with a single sgRNA gene or the pools of 3?
Yeah, good question. Thanks. So this is so when going to the multiplexing, we dialed it into the best performing individual guide. So we suggest starting with the pooled guide approach and coming in and starting with those pools and then experimenting with them individually and dialing in the best performing individual component, and then using that as part of the multiplexed pool approach.
Great, thank you. So our next question, does the CRISPRi guides share the same sequence of the Horizon CRISPR-A off-the-shelf products?
Oh, right. Yeah, good question. Thanks for that. They're not. So they have to nest on sort of opposite sides of the transcription start site. So the CRISPR-A guides are just upstream, and the CRISPR-I guides sit just downstream of that to kind of have the right actions on transcription. So so they're close. They're neighbors, but no, different targeted areas across the genomic DNA.
Great. Thank you so much for that clarification. And so, Steve, it looks like we may have time for one more question before we close out this webinar. So how far from the TSS is the gRNAs need to be designed to achieve gene-significant knockdown using this system?
Yeah, so related. They're just they're within like 300 base pairs downstream, so they're nested very close to that transcription start site. And again, this comes out of the published algorithms from the Wiseman lab.
Wonderful. And it looks like we've got another question in here for you. Do you recommend only single guide RNA rather than a 2-part CRRNA or tracker RNA?
Yeah, sure. It's a good question. For this system, we were using the single guide RNAs. The 2-part, the CRISPR tracer RNAs would work in this system, but we're the only guides we're offering are the synthetic single guides. We've tried it a bit in passing. It seems that the market is moving to the synthetic single guide more and more, so we've leaned on that for this product menu. But we have both across our other products, and we see quite similar results across them. So yeah, sure. If there was a reason that was the better option, then yes.
Great, thank you so much, Steve. Do you have any final comments for our audience before we go for the day?
No, thank you. Thanks for all the great interaction and great questions. Please feel free to reach out to myself or our tech services group. Again, we'd love to help you get going with CRISPRi. Super excited about this and just to see what comes of it in the months and years to come. It's been a fun development, and it's great to see it go out into the wild today. So yeah, we'd love to hear more from you, and please feel free to email or call us anytime and talk more. So thanks Alexis. Appreciate the hosting, and thanks everybody for joining.
Thank you again, Steve, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Horizon Discovery, for underwriting today's educational webcast. Before we go, I'd like to thank the audience again for joining us and for their interesting questions. As a reminder, questions that we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information that you provided at the time of registration. So please feel free to keep those questions coming in. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. Stay healthy, stay safe. Thank you, everyone. Bye-bye.